Okay, I would like to begin by thanking the organizers for giving me this opportunity of sharing some of our work with you, and also for enlarging the discussion of complex networks to include the sorts of things I'm going to talk about. So this work has been done with several generations of students and postdocs whose names I have up here. They're all over the country now, or the world, and these are my current students. And this is our good friend from the University of Ghent. And I would like to acknowledge support from various funding agencies. We publish more than a few papers on this subject. I will not inflict all of them on you, but I'll give you a colloquium-style talk on this subject, which doesn't mean I leave out all details, but I won't have all the details there. And if you're interested, I'll be happy to discuss them with you in the lunch break or other breaks. So our initial motivation was to study spatiotemporal chaos and its control in arrhythmias. Actually, we were studying some related models in the old days. And then we have looked at problems in which are more and more details about cardiac tissue. There are words which, if you're like I was a few years ago or a few decades ago, you will not know the meanings of. Fibroblasts, Purkinje fibers, so on. So the purpose of this talk is to introduce you to some of these terms and the mathematical models that are used to describe wave propagation in cardiac tissue, and of course the multi-billion-dollar question of how to suppress cardiac chaos. So we've done many simulations. Let me not give you a list now. Let me just proceed. Cardiac arrhythmias, like ventricular fibrillation, are a major cause of death in industrialized countries, and on this scale, India is industrialized. As we will see, ventricular fibrillation is associated with broken spiral waves of electrical activation on cardiac tissue. So the goal is to understand the dynamics of ventricular fibrillation in the presence of all sorts of tissue heterogeneities which always exist in cardiac tissue, and eventually to develop low-amplitude defibrillation techniques. What that means, I will tell you as we go along. All of you must remember these sorts of schematics of the human heart from your school, if not some other place. There are 4 chambers: the right atrium, the left atrium, the right ventricle, and the left ventricle. At some level, the left ventricle is the most important, insofar as it pumps oxygenated blood to all parts of the human body. And in addition to this sort of thing, with all its valves and so on, which you know well, there is the electrical system. Here is the sinoatrial node, which is the pacemaker of the heart. It sends electrical signals, which are carried by special conduction pathways. There are all sorts of things, bundle of His and so on, and also the so-called Purkinje fibers, which I will get to presently. And, this should work well, because as you know, the mammalian heart is one of the most efficient electromechanical devices. In humans, there is an average of 72 beats per minute for an average adult, about 2.5 billion beats in an average lifetime. That's really impressive. And this rhythm is maintained by the electrical activity of the heart, in particular the sinoatrial node. But if some patient gets these unfortunate cardiac arrhythmias, namely ventricular tachycardia or ventricular fibrillation, then there is trouble. Here is an electrocardiogram taken from a patient who had it all strapped up. These are so-called Holter tests. So here all is well, even to the untrained eye. That's the so-called normal sinus rhythm. And then it speeds up. Tachycardia, which is basically Latin for fast heartbeat, even to the untrained eye, that's a fast heartbeat. And then something even worse happens here. Even the untrained eye can see that it's not only fast, but it's also disordered in time, and that is ventricular fibrillation. And in the absence of medical intervention, from here to death is 2.5 minutes. That's all the time you have to act. It is now more or less the general consensus that such ventricular arrhythmias are associated with the formation of spiral or scroll waves of electrical activation on cardiac tissue. This is from some canine heart where it was artificially induced and viewed with voltage-sensitive dyes. And I have an animation here taken from the web page of Cherry, and you can see this is the normal heart rhythm. That's the plane wave from the sinoatrial node. Ventricular tachycardia is associated with a single spiral wave, and let me just play it one more time. And ventricular fibrillation is associated with the broken spiral waves, and here are actual measurements in tissue, okay? So you can't see that very clearly, but this is the phenomenon that we are after today in today's talk. So cardiac tissue is excitable in a technical sense. That is, if there is a subthreshold perturbation, it dies away, but if you perturb it above some level, then the cardiac cell, called the cardiac myocyte, gets excited and then cannot be excited anymore during the so-called refractory period. So here is a typical action potential. Actually, this is from some mathematical model of the type that I will describe presently. That's the resting membrane potential around minus 85 millivolts. And here it's excited and it goes like that. And this is the depolarization associated with a fast inward sodium current of sodium ions. Then a plateau because of slow inward calcium currents, and repolarization because of outward potassium currents. Typical time scales, well, 200 to 300 milliseconds, all right? So it's quite different from the numbers you see in, say, brain cells. Now, what do the models look like? By models, I mean mathematical models, not the way in which a biologist uses the term model. So they're really reaction diffusion equations. V is the transmembrane potential. I is the current density in cardiac tissue. C is a capacitance density. Here is a term that allows for coupling between different cardiac myocytes. And at the simplest level, we can think of D as a scalar. But of course, in real cardiac tissue, it's a tensor. And I will get to that later in the talk. There are many, many types of models. The simplest one are Fitzhugh-Nagumo type models. We use the one associated with Panfilov. And then there are more and more with greater and greater realism. This is the Tentuscher Noble Panfilov model for human ventricular tissue. Another one, Tentuscher and Panfilov, even with more recent measurements. That's from 2006, human. And this is the O'Hara-Rudy, even more recent. These keep on developing as measurements keep on becoming more sophisticated. But let me show you what they look like. First, the simple two-variable model, the Panfilov model. Here, I'm sorry, I have E instead of V just because of the original terminology in the paper. So E is the transmembrane potential. This is what it looks like. So this is the transmembrane potential. And all information about refractory periods, etc., are buried in this coefficient, which goes with this slow variable G. Okay? But even at this level, it's certainly not a very pretty partial differential equation. It's not as elegant as a deceptively simple Navier-Stokes equation. Here, this f of E is a piecewise linear function which takes several inequalities to specify, which of course I will not go through in detail. And then there are numbers. These numbers are picked so that the action potential that we get from this model is in the right range of parameters of the sort that I showed you earlier. Now, okay, you can solve it on a computer if you're a bright graduate student. And okay, I won't give you details, but we know how to do these things. And if you do that, then you will find in some region of parameter space a state which clearly has broken spirals. And for the rest of this talk, this is the mathematical analog of ventricular fibrillation. Okay, and let me show you what it looks like. All right, so here we go. Oh, what happened? Doesn't like it. Should work. All right, at least it's working in that corner. All right, so that's the spiral wave, and it's going to break. And you will see that when two waves collide, they don't go through each other. That's characteristic of waves in such media, because the region behind the other wave becomes refractory. It cannot be excited. So the waves don't go through each other, they just annihilate. Okay? But, you know, you can get this sort of state which has turbulence in it. This is what causes ventricular fibrillation. This is the leading cause of death in the industrialized world. So it is very important that we learn all its properties and learn also how to eliminate it when it does arise. Let me show you a little more biologically realistic model. You know, the sort of model I wrote down is probably all right when you're talking to physicists, but if you want to be taken seriously by a biologist, you have to have a more realistic model. And so I just give you, you know, please forgive me, but I have to do this just to show you what the model is like. So I'm showing you the TNNP model. This is biologically realistic because it incorporates details of ionic currents, ion channels, ion pumps, ion exchangers. If you don't know all that, I mean, okay, we can discuss it later. This has 12 ionic currents, 12 gating variables, intercellular sodium, ions, potassium ions, calcium ions, and their concentrations in the cytoplasm and the sarcoplasmic reticulum. So again, basically a reaction diffusion equation, which you might think is very nice and very simple. It almost looks linear until you ask what is in this current, and the subscript TNNP just refers to the model. Well, this current is a sum of several currents from ion channels, ion exchangers, and so on, each with its own subscript. And the inward currents are because of sodium and calcium, the outward ones because of potassium. And then there's the Nernst potential, which gives you the potential at which inward and outward currents are balanced. And then every such ionic current follows a sort of Ohm's law, a conductance times Vm minus this Nernst potential. And actually, in addition, we should put in some probability of the ion channel being open. So I've indicated that by epsilon. Okay? And then each one of these gating variables, as they are called, there are 12 gating variables here, satisfy ordinary differential equations of this sort. So any generic gating variable epsilon satisfies this sort of an ordinary differential equation. And then these alphas and betas are the rates at which the gates open and close. Okay? And you can solve for the asymptotic state. How do you get these alphas and betas? Well, you get them from measurements. These are so-called patch clamp measurements. And they depend in a very nonlinear way on the membrane potential V. Okay? It is so complicated, I'm not going to write it down and refer you to any one of the standard papers. And they use units which are typically unknown to the standard physicists, and it takes a while to get to know all the units used in electrophysiology. But once you have done that, and you're a bright graduate student, you can put it on a computer and try to solve it. Okay? By the way, it's such a complicated set of equations, if I actually wrote it down on paper, it would take me about 10 sheets. All right? So I'm not putting it up. And eventually you will, for those of you who are real experts, you have to have boundary conditions. Typically you use Neumann boundary conditions because atria are insulated from the ventricles and so on. Eventually, to solve such equations, you have to go to a cluster computer, and believe me, that you can get it to look like a good parallel code, which I will not describe to you, but my students are very good at. And then how do you initiate a spiral wave? You pass one wave from one side, wait a little bit, pass another plane wave perpendicular to it, and then a spiral forms, and you get a wave, okay? So I'm showing you for different parameter values the development of spiral waves. This is the case where it develops into a single rotating spiral. And since there is some sort of pedagogical aspect to this workshop, you can see that when there is a single spiral, if you take the time series from a representative point, you get oscillatory behavior, which if you take a power spectrum of, you will get nice, well-resolved peaks at harmonics, and that's periodic behavior, okay? But that is the mathematical analog of ventricular tachycardia. However, if the spiral wave breaks, then you get much more complicated time series, and there's a broadband background in the power spectrum taken from the time series at a given point. All right, so you can use all your nonlinear dynamics types of analyses here to characterize these states. All right, and you can get multiple spiral turbulence and so on. So instead of, come on, why is it taking so long? Fine. All right. So this shows you how the spiral wave develops for different parameter values. I'm sorry for the interruption in the flow of the movies. And so this is how a realistic model shows you spiral wave breakup. I first showed it to you for a simple two-variable model. This is fairly current, ionically realistic model, and this is how the spiral wave develops. Okay? All right. So now, the billion-dollar question. It's a multi-billion-dollar industry. If a poor patient does get such a cardiac arrhythmia, there are two options. One is pharmaceutical means you administer some medication, but normally, that's not fast enough because, as I said, from initiation of ventricular fibrillation to death is 2.5 minutes. So all of you must have seen Hollywood movies where they come along, a patient falls, and they come with paddles, and they give an electrical shock, and on good days it works, and all's well. And then in high-risk patients, they actually put in so-called internally implanted defibrillators, which sort of look like this. You can do an internet search and find one of these objects, and it is, roughly speaking, planted like this. For example, here it is planted here, and then there are these catheters that go to the heart, and they monitor the state of the heart, and if all is well, they don't do anything. But if it looks like something bad is about to happen, that is, some time series looks bad, they give a large shock, and again, on good days it works. However, I'm told that it feels like being kicked in the chest by a bull. And in the process of setting everything right electrically, it can lead to some damage, the formation of scar tissue, which then becomes a site for new arrhythmias. All right? So there is a great need to develop low amplitude defibrillation schemes, and we worried about this the early 90s, and we came up with a scheme which does the following. What you didn't notice in the movies, because I went through them quickly, is that the spiral waves were being absorbed by the boundaries because we were using Neumann boundary conditions. So we said that we know what the spatial organization is of these broken spiral waves. If we excite the tissue along some grid, a mesh of electrodes, then, because that will make the tissue refractory, it is as if you will be imposing Neumann boundary conditions on small patches of the simulation domain. Here, there is the evolution without any control, and here there is evolution with the control. You can see the grid along which we have applied a current, only for about 1,000 milliseconds, and that's enough to absorb all the spiral waves and destroy them. So let me be conservative and try to run this simulation in the following way. So you can see there that the spiral wave is completely absorbed well within 1,000 milliseconds, which is much, much smaller than 2.5 minutes. So our numerical patient at least has been saved. Now, of course, you might say, does it work when I have a bigger region? Answer is yes. Does it work in an ionically realistic model? The answer is yes. So I give you a simulation for the TP06 model. And here again, you see for all types of spiral waves, broken or not, our control scheme works pretty well. In 3D, it is not so wonderful because, of course, if you had a 3D mesh, you could do it, but you don't want to cut into cardiac tissue in the third dimension. You, I guess, want only to put it on the wall, the inner wall, or the outer wall, but most probably inner wall. And what we were able to do by some clever pulsing, which I will not explain, is at least a thin slivers of cardiac tissue, we could eliminate such spiral wave, or rather a scroll wave in 3 dimensions by suitable pulsing, but not for biologically realistic thicknesses. In any case, one of my colleagues forced us to apply for a patent, which we did, which was eventually granted, and my then students and I are named as inventors. But industry in this trade is very conservative. They only want to use what they have, and they won't talk to you unless you've done human trials, or at least animal trials. So that is something of the future. But all the time since then, we have been refining our studies to see whether this scheme of eliminating spiral waves, which are the mathematical analog of ventricular tachycardia or ventricular fibrillation, is robust enough as you increase more and more realism. So first, let's look at inhomogeneities and obstacles which can occur in cardiac tissue because of scar tissue, which has been formed earlier. And there were a variety of experimental results around that sometimes such inhomogeneities can lead to fibrillation. Sometimes they can, suppress it partially, make ventricular fibrillation go to ventricular tachycardia, or completely suppress ventricular fibrillation. And experiments are done. For example, here, this is chicken cardiac tissue to make an inhomogeneity. You just punch a hole through it. Or you can do it in vitro, where you lay down cardiac myocytes and put as many obstacles as you want of any type. So we, of course, can introduce obstacles in our simulations. And indeed, we find that sometimes the inhomogeneity can cause spiral breakup. Sometimes it suppresses it. It can even suppress it completely. The red, blue, and green is a color code. And we showed by extensive simulations that what you get, whether you get suppression or not, depends very sensitively on the position of the inhomogeneity. It is, if you like, a spatial analog of the sensitive dependence on initial conditions. There is sensitive dependence on the position of the obstacle. Okay? These pictures, which I'm not going through in detail, show you that. And in fact, we worked out a stability diagram in the simulation domain. So if your inhomogeneity is here, then you still have spiral turbulence. If it is in the blue region, you get a spiral anchored at the inhomogeneity. If it's in the green region, the spiral turbulence goes away completely. So this is, then we checked it in more and more realistic domains. So this is a simulation in the 3-dimensional TNNP model with actually muscle fiber orientation. So now the diffusion constant is no longer a simple scalar. It is a horrible tensor, and the components of the tensor have to do with the way in which the muscle fiber cells are oriented. And here again, qualitatively, the same message. Sensitive dependence on the position of the inhomogeneity. Here is something in an anatomically realistic domain. This is what they never teach you in mathematical physics when you study mathematical physics. you have very nice boundary conditions, periodic boundary conditions, or some nice boundary conditions in a square or something. This simulation is done in an anatomically realistic domain. How do we get an anatomically realistic domain? That's because very, bright experimentalists measure these by using something called diffusion tensor magnetic resonance imaging. Not only do they, measure for some given mammal the type, shape of the heart, but also the muscle fiber orientation. Okay? So you can study these in many, many different ways. Let me just show you a few movies to tell you what sorts of things we get. So here you can see two inhomogeneities, one square here, one there, and this spiral wave is anchored to both of them. It jumps from one to the other, but really it just goes round and round. So it is, ventricular tachycardia, if you will, in a simulation domain with two inhomogeneities. Let's look at a broken scroll wave in an anatomically realistic domain. So that's an anatomically realistic domain, and here is a scroll wave which is sort of breaking, not completely. But, this is in a simple model, the Panfilov type model. Can we do better? Can we look at other inhomogeneities? The answer is yes. There are certain inhomogeneities which are not conduction inhomogeneities. So far, the sorts of inhomogeneities I talked about, is conduction inhomogeneities, the signal just doesn't go into that region. There's some scar tissue or something. But you can have ionic inhomogeneities where, because of some particular condition, let's not go into details, the conductances of the ion channels in a given region are different from their normal values. I will call that an ionic inhomogeneity. And we have studied those too. And you can get very complex spatiotemporal behavior. For example, now the wave can go into the region of the inhomogeneity, and you can find cases where the behavior of the wave is spatiotemporally chaotic inside the inhomogeneity, but periodic outside it. So I'm not showing you every detail, but suffice it to say that mathematical models for cardiac tissue, if they're sufficiently detailed, of the sort that I have given you, can study fairly realistically all such things. You can ask, does this mesh-based control that I talked about work when we have such ionic inhomogeneities? The answer is yes, and I can show you one animation, okay? And you see there's an inhomogeneity which is acting more or less like a conduction inhomogeneity. Up in these two things, there is no control. Here we have the grid, and we have managed to control the spiral wave turbulence even in the presence of an inhomogeneity. All right. Now, cardiac myocytes are not the only cells that are found in cardiac tissue. There are certain cells called fibroblasts, which are not excitable in the sense that they won't display an action potential if you excite them in some way, but we can put them into models. Fibroblasts proliferate in many cases. Sometimes they proliferate in the process of wound healing, so in following a myocardial infarction, or they just increase with age. All right, so what do they look like? You can't see very well here, but these experimentalists are really very, very clever, and they can stain tissue in some way. And over here, F, these are the fibroblasts, these are the myocytes, fibroblasts. I think the top one is in the sheep ventricular myocardium. This is the rabbit. And the organization of fibroblasts in myocytes is not something which is a settled issue. We can't say this is the model. So when you study mathematical models, in addition to the one that you had for the myocyte, which is M, you have to have equations for the fibroblasts. I'm not going to the details, but you can ask, what sort of model are you going to use for the spatial distribution of these fibroblasts? And we have used three different types in our group. One is a bilayer, where the black cells here are the myocytes, and the gray ones there are the fibroblasts. and they're connected by so-called gap junctional couplings. And if you look at details, the sizes of fibroblasts are considerably smaller than the sizes of myocytes. And you can attach more than one fibroblast to the myocyte. So that becomes another parameter that you can and do vary within these simulations. You can say, well, perhaps they're actually interspersed randomly, like, those of you who do percolation or random resistor networks, you can randomly remove myocytes and replace them by fibroblasts. We have looked at those types of models too. And so on, and biophysically realistic ranges of parameters, which I have listed here, but I will clearly not discuss. And then you can ask, what happens? How do these fibroblasts affect the spatiotemporal evolution of the spiral waves in these, in these simulations? And can we also look at fibroblast inhomogeneities and so on? So I have here some simulations showing a simulation with no fibroblast inhomogeneity, and these ones with different types of parameters and so on. And sometimes we find, I won't show you every movie, that fibroblasts, depending on parameters, can act like induction inhomogeneities, where the signal does not go in, but for other parameter values, they can act like ionic inhomogeneities, where the signal does go in. But even with that case, we can suppress the cardiac chaos when it does occur with our simulation scheme. If you have randomly distributed fibroblasts, which is the second type of model, you see this sort of, this crenellation on these waves. That's because of the random distribution of fibroblasts, and depending on the type of randomness, either the signal stops propagating, or you can still sort of identify a single spiral wave or multiple spirals and so on. And at the end of the day, what you discover is, you can do this in 3D too, which I'm not showing you because of lack of time. As you increase the concentration of fibroblasts in these random models, then initially you can have a single rotating spiral, and then you can have a very complicated transition from single rotating spirals to multiple rotating spirals, which are periodic or quasi-periodic, etc., then spiral turbulence, and eventually, if there are enough of these fibroblasts, then no wave propagates, and the spiral is absorbed completely, okay? And you also find this in 3 dimensions. So this is, if you like, a very nice example of wave propagation in a random medium, a medium of great concern to all of us, namely cardiac tissue. And then, again, I will not show you the details. You can eliminate it, at least in 2 dimensions, with our control scheme. There's an important problem which I will not dwell upon too much here, but I at least want to tell you about it. So far, I've been arguing as if the mechanical system is not important. If you're careful, you will say the mechanical system of the heart, namely it beats, so it expands and contracts, is enslaved to the electrical system. My equations have so far been looking at the electrical system. But really, you should couple both. And there are simple models of the type we have studied, which I'm not going to explain. But there is some important biophysics also. There are so-called stretch-activated currents. So how much current goes through a given ion channel depends on how much it is stretched. And people are beginning to study those. All I want to say is that we've done some version, and even with that stretching, our control scheme works at least in 2 dimensions. I think, okay, I have 4 minutes left. We can model these Purkinje fibers too, which I showed you. They're the specialized conduction pathways that help in taking the electrical signal from the sinoatrial node down to the ventricles. Okay, let me not show you everything. Perhaps I'll just show you one movie because it looks nice, where we can put these, Purkinje fibers in some way. There really seems to be a fractal network but nobody really knows. Some people try to, this is how it evolves. You see that fractal network, that's the Purkinje fiber, and we can study in great detail how the layer of Purkinje fibers communicates with the layer of myocytes. So this is sort of like the model I told you, a bilayer model, where we have a Purkinje fiber layer and a layer of endocardial cells, and there's communication by the couplings between these layers, and if you have a good enough model, you can study such spatiotemporal evolution, at the level of these models. But again, I think there is no hard and fast thing. There is no given model. The measurements are still not good enough to tell you precisely what model you should use for the spatial organization of Purkinje waves, Purkinje fibers, excuse me. Now let me show you some more realistic simulations. I showed you some baby simulations on some anatomically realistic things. So this is a scroll wave in a dog heart model, the Hund-Rudy model, which is very complicated to write down. So let me show you one where there is a single scroll wave. This takes a lot of time on the petaflop computer that we have, for those of you who like high-performance computing. And then if you change parameters like conductances and so on, you get a broken spiral wave. This is again in a dog heart model. Okay. And then you can ask, well, what happens in the human heart? So this is the TP06 model I told you about. This is a single scroll wave in that anatomically realistic domain. It's a very short movie, I'm sorry. And then you can also get a broken wave. And that is, again, in the human thing. If you run it long enough, you see it's breaking already. So the eventual goal is to understand the spatial temporal dynamics of such waves in such anatomically realistic domains and come up with a scheme where you can get low amplitude defibrillation, but also to examine what other arrhythmogenic factors there are. For example, we have found that a gradient in the density of fibroblasts can also be arrhythmogenic, and this is in the O'Hara-Rudy model, which is really a state-of-the-art model. And here you can see the formation of a scroll wave. Perhaps you can't see it very well, but I think you can see something scrolling over there. Probably in the next movie you can see it better because the color scheme is better. And while you watch that, then let me stop and say that, if you are willing to look at detailed mathematical models of the sort that I have put up, you can study in quite great detail different types of cardiac arrhythmias in cardiac tissue. You can examine in great detail, much greater detail than is possible in experiments, in vitro or in vivo, of the variation in different properties like conductances, the amount of fibroblasts, the coupling with the Purkinje fibers, and so on. So in this field, I would like to say that certainly numerical simulations of such realistic models is becoming as important as in vitro and in vivo experiments. Thank you. I'll be happy to answer questions if you have any. Yes? Okay, that's sufficiently technical that let me answer that question separately.
Okay.
I will give you the reference to our paper and then you will get every detail that you want.
Thank you, sir.
Certainly it's not something that, okay. Yes? Well, cardiac arrest is probably caused by this. This field is more than 100 years old. I think the oldest experiments go back to Mind, who was in the university in Montreal. And in fact, he probably passed away in his lab. So it was well known to such pioneers that if this arrhythmia, namely ventricular fibrillation, sets in, cardiac tissue starts sort of quivering. And if let's say the left ventricle of patient A goes into this quivering state, then it becomes incapable of pumping. So once the heart in that state, it cannot pump blood. Once it cannot pump blood, then the brain is starving of blood. 2.5 minutes, no more. So I think that is cardiac arrest. But okay, whether the cardiologist, more technical definition, I don't know. But roughly speaking, yes. Sudden cardiac death is caused by this.
Rahul, I got a bit confused. So, where you had that grid of inhomogeneity which was controlling your spiral wave, how dense, I mean, how closely do you have to play?
Very close, actually.
Okay.
We've tested many things, and it's pretty...
But it will still scale with the system size or something? I mean, suppose you had a...
You see, in fact, the main criticism of some of these simulations is the system sizes are too big for human cardiac thing. I mean, roughly speaking, that's the size of your heart.
Yeah, right.
Now, let's take some sort of pair of scissors and cut it open and spread it out. It's 5 centimeters by 5 centimeters. The simulation domains we have are more like 9 centimeter by 9 centimeter. That's partly because we want to get the spiral breakup. If it is very tiny, you don't get such spiral waves, which is consistent with the observation that small mammals do not get such arrhythmias. Unfortunately, on that scale, humans are large mammals.
I also have a question.
I come to you, yeah. No, go ahead, go ahead.
I also have a question about control scheme. I would like to clarify. What is the coordinate of pushing this stimul 100 millivolt? This is a centrum of vortex?
No, no, doesn't matter. We don't have to keep track of the position of the vortex. You are right that in the case that there is a single rotating spiral, some groups have, paced only at the core of the spiral and use that to drive the spiral out of the simulation domain. The important quality of our control scheme is that it works even when the spiral is broken. Those schemes will not work when the spiral is broken. Okay? And the broken spiral state is a much more dangerous state. That is ventricular fibrillation.
And to where exactly you...
No, we put it on a mesh.
On whole
mesh.
Okay.
But it's not global. Global ones can also work. Okay. Roughly speaking, the current thing is global. You apply the voltage everywhere. But what we're trying to say is because we know a little bit about the spatial temporal organization of these spiral waves, we can use a mesh, and it's a fairly, not a dense mesh. Okay? And that's enough. In fact, we've even checked that it just works with lines. You don't even need the second thing.
And this depends on the size of vortex. It should depend on the size, no?
Not really. Not very much. I mean, actually, if it's a single spiral, it's quite easy to remove. It is the broken spirals which are hard to remove.
Thank you.
Yes. It could be, of course, because, even more important when there are inhomogeneities, because spirals can and do get anchored to inhomogeneities, one of the simulations I showed. So, in an anatomically realistic domain, if you have some inhomogeneity, the chances are that if there's a single scroll wave, it will get anchored on that, unless it breaks.
So the grid-based spiral scheme, the scheme you use to control the abnormal behavior, what is the effect of that on the normal behavior when the normal waves are?
That's right. So, of course, it would also eliminate this plane wave, but remember, we keep it on only for 1,000 milliseconds. So what is not in the model is, we haven't put in the sinoatrial node into the model. The assumption is that once you remove this, then the sinoatrial node takes over, does its job, and all is well, and it happens. Okay? Yeah. Good. Then thank you very much.